Hello, everyone, and thanks for joining us. Appreciate you hanging in there to our the last afternoon of our symposium. This is session H on exposures and health effects. We have a great lineup of talks from Chi Lee at University of California, Riverside, Olivia Ryder from Sonoma Technology, Andrew Patton from Johns Hopkins University Bloomberg School of Public Health, and Ranga Thiruvenkatachari from University of California, also from University of California, Riverside. So thanks again for joining us, and please, as questions come up, please type them in the Q&A area so we can follow up at the end of each presentation. So Chi, take it away.
Thanks, Mary. So good morning, everyone. My name is Chi Lee. I'm a PhD candidate in environmental engineering from CSER, UC Riverside. Today, I'm going to talk about my recent study on investigating the health effects of particulate matter on mice by using the animal exposure chambers. On the background, pictures showing the dual whole body exposure chamber that we have right now. So let's start with some brief introductions. What particulate matters that we are interested in and we have studied? First of all, we have studied the salt and sea dust and the water spree. The reason why we think it is meaningful to study salt and sea nearby dust and water spree is because the salt and sea itself is currently undergoing a rapid retreat. It is exposing dry lake bed, which is also known as a playa, and as a result, it's producing increased dust. Meanwhile, we found the water over there is getting more and more hypersaline, causing a dead offs from birds and fish, and together this caused a relatively bad air quality in the area. We also found the highest rate of asthma development rate around the over the communities at the area, and this is indicating a potential connection between those two. We also studied the swine dust, which is believed to be a potential source for particulate matter that can trigger the lung inflammation and even asthma. We studied Alternaria, which is a known allergen. They are suspected to cause strong inflammatory response. We studied ground silica. We use it to simulate the desert dust, but meantime, we try to eliminate the effects from the soluble components. So the ground silica itself is an inert large particle. Okay, so here is a brief schematic of our system. The chamber system contains three main components: the aerosol generation system, the exposure chamber, and the aerosol monitoring system. There are different ways to aerosolize a liquid. Right here, we use the compressed way. We use the nebulizer for most of our particulate matters. We also use the Ponder dispenser to generate the particles from Ponder, usually used for large particles. For example, we use it for the ground silica, which have a peak size around 1.5 micrometer. For the aerosol monitoring system, we have the SMPS, which is obviously give us the size distribution, and we can calculate the mass concentration from it. We also have the relative humidity and temperature probe and ammonia analyzer to help us record the status of the chamber. So here is a brief workflow of how we usually, how a typical whole body exposure chamber study looks like. So when we have the mice order in, we will have the mice settle down for 2 weeks before any experiments. And then we will move the mice in the chamber for either 7 days or 28 days exposure. After exposure, the mice will be moved out for long lavage and further dissections. And then it's the time that the different medical labs are going to come in and collect different samples from the mice, including the lung samples, gut samples, brain samples, spleen samples, and so on. And then they will take the samples back to their own lab and do further analysis, including the immune system response, gene expression and modifications, and so on. So my primary work in this study is to make sure I can provide a stable particle aerosol environment for the mice. To keep the consistency through different exposures using different type of particulate matters, there are two factors that must be controlled in our study: the PM mass concentration and PM size distribution. To keep the consistency is to ensure the introduced doses of PM to the mice are similar and can only be affected by the mice activity. And to be more specific, the reason why we want to control the mass concentration rather than the surface area concentration is because the most of the PM used in our study were water soluble. And the reason why we want to control the size distribution is because the particle deposition friction shows a high dependency on particle size. The figure on the left here shows the predicted particle long deposition friction as a function, vary as a function of a diameter. This is from the MPPD model. We also did some studies to prove this. We here to give an example that we did about our particle long deposition studies. So over here, we let 3 mice exposed to the 1 micrometer fluorescent beads for 3 hours. And on the top left figures, we're able to achieve a total number concentration around 800 per cubic centimeter. And if you look at the second figure right beside it, we're able to achieve a monodisperse. And overall, from the images that collected from taken from the mice lung slice, we see the 1 micrometer beads particles can reach alveoli and are quietly, evenly distributed in the alveoli. So to keep the consistency, we also need to control something else. In the next slides, I'm going to show you the ammonia data collected from 28 days alternaria exposure and CO2 and relative humidity data collected from 28 days salt and seawater spree exposure. So first, let's take a look at the ammonia data. First of all, you can see a clear weekly pattern, right? The ammonia mixing ratio stayed pretty low, below 300 ppb for the first 2 days, and then went up exponentially in the next 5 days and reached 13 to 41 ppm by the end of the 7 days. And the reason why we see this, the pattern repeat every week is because we do chamber cleaning every week. We change the bedding and we change the bedding, we change the food and water. We can also see a similar NOx ammonia mixing ratios in the exposure and the control chamber, in which is indicating the primary source for ammonia is not from the aerosol stream, is actually from the mice itself. By doing the weekly cleaning, we're able to achieve a weekly average the ammonia concentration below 25 ppm. Here I'm showing the CO2 and RH relative humidity data. We can also see a clear pattern of CO2 mixing ratios that repeats every day and night. This is indicating a normal mice living activity. We're able to control the relative humidity at a good amount, around 40%. So here I'm going to use the salt and sea study as an example to introduce the typical whole body exposure studies. So the figure on the left here shows the PM mass concentration over a 7-day exposures. We use, in this specific study, we use alternaria as a known allergen and to set up a reference for typical lung inflammatory response. we use a Pacific Ocean spree as a control group to compare to the Salton Sea exposure, which is obviously the objective in this study. We're able to keep this stable aerosol suspension in the chamber for 7 days and with a PM mass concentration around 1.5 microgram per cubic meter. The figure on the right here shows the average size distribution of those 3 different types of PMs. You can see I'm able to achieve a similar size distribution with a peak diameter within 79 to 95 nanometer on average. I also did a particle chemical composition analysis by using the aerosol mass spectrometer. So from the figure shown on the left, you can see the alternaria here has the highest organic friction of 82.3%. It has very little inorganic species or metal, and the alternaria is the only one that has notable friction of ammonia species. We know that Salton Sea sample has higher salinity than the Pacific Ocean sample. Salton Sea, and in the AMS analysis, we able to make sure that the Salton Sea has more sulfate than the Pacific Ocean. But in the later analysis from the medical side, it is indicated that the sulfate itself does not have obvious effects on the mice. We see that both Salton Sea and the Pacific Ocean water shows a seasonal variation in terms of the chemical friction. We see higher organic frictions in the new collected samples in September and October. So let's go to some medical data. So from the medical side, we see this strong inflammatory response from the alternaria exposure as expected. And while we don't see the Salton Sea exposure generated an equal inflammatory response similar to the alternaria. However, I'm sorry. I will also see this strong gene expression change observed in the alternaria exposure, which is consistent with the histology images that I showed in the previous slide. However, we don't. We also see the Salton Sea exposure was able to trigger an inflammatory response, but differently than the alternaria exposure. A shows high expression on the gene analysis that shows a high expression on genes associated with asthma. And as expected, the Pacific Ocean spree shows negligible response in neither the lung nor gene analysis. With that, I would like to thank my 2 advisors, Dr. Lowe and Dr. David Colger. And also I want to thank all my colleagues that helped me in this project. And this researcher was supported by the National Institute on Minority Health and Health Disparities of the National Institute of Health. And if you're interested, you can see scan the QR code here and take a look of our previous publications. And thank you.
Thank you very much, G. If anyone has a question, please feel free to type it in the Q&A, and I will ask just, so what do you think are the next steps based on what you found? What would you like to look at next?
So as I introduced in the result session from medical side, we were able to find this kind of special response from the mice in the exposure of Salton Sea spree. It is not able to introduce a lung inflammatory by itself, but meanwhile it is able to trigger some gene expression that associated with asthma. So the next step we are planning to do is we are going to do a salton sea exposure similar to this one first, and then right following it, we will do an alternaria exposure at a lower concentration. So we think that the salton sea is still a gas currently. We need to make sure about this, but we think the salton sea water may somehow increase the rate to develop asthma. Yeah, that's the next step.
Great, that's really interesting. Yeah, yeah, I mean, it does seem surprising, right? You don't see the inflammatory response, but yeah, there's definitely something going on there, right?
Right. Yeah. We'll see.
Great. Well, thanks so much.
Thank you.
And if any questions come up as we continue through the speakers, please feel free to type them in, and we can either respond just in the Q&A, or we'll circle back and speak about them later on. But next up, we have Olivia Ryder. Olivia, are you ready to go? I see your slides. Olivia, are you there? Okay, looks. I see Olivia is having a couple of technical issues. We'll give another minute to see if she can get her mic issues sorted out. So why don't we go ahead and ask Andrew to get set up, and we will circle back to Olivia. Oh, okay. Hi, Olivia. I can see you. Are you?
Hi.
Is your mic on? There you are. Okay. Very
Sorry
good.
about that.
No problem at all. Take it away.
Okay, thank you so much. Those are in presenter view. Perfect. Yeah, thanks so much, Mary. So today I'll be talking about some results that we have from an investigation that we did into ambient near-road mobile source air toxics in the United States between 2016 and 2018. And I just wanted to acknowledge the funding for this work came from Texas Department of Transportation. And I have now, let me see, there we go. So the gas phase mobile source air toxics, or MSATs as I'll probably be referring them to them as, include 1,3-butadiene, acetaldehyde, acrolein, benzene, ethylbenzene, formaldehyde, naphthalene, and polycyclic organic matter. And so that defines a broad class of compounds that includes polycyclic aromatic hydrocarbon compounds, and that includes benzo-A-pyrene, which is one of the species that we will be looking at in this study. And so prior to 2014, these MSATs actually hadn't been routinely monitored next to roadways in the United States. But in 2014, EPA implemented the Near Road Monitoring Network, and so that mandated that these types of measurements take place across the country. And so between 2016 and 2018, one or more of these species were measured at 17 monitoring sites in the United States that were within 50 meters of a major roadway. And so that was part of the broader network of near road monitoring sites that was also monitoring criteria pollutants. And so on this map, what I'm showing is where these 17 sites are located across the country, and also which species were being measured at which of the monitoring sites, to give sort of an idea of the spatial variability. And so the reason that, of course, we're concerned with these levels of species is their health impacts. And so 7 of the 8 air toxics that I've listed on the slide are also part of the US EPA's list of 30 urban air toxics that pose the greatest potential health threat in urban areas. And so those ones I have bolded to make them easier to see. And so the questions that we came in to ask, well, first of all, we investigated the ambient concentrations. Oh.
Great, thanks, Olivia. Can you go ahead? Hi, Olivia. We are seeing your slides, but we can't hear you.
Hearing me? How about now?
Yes, we can see you and hear you.
Okay, sorry about that. I got kicked out somehow. I'll just pick up where I was. Did you hear everything else that I said, though?
Yes, we did.
Perfect. Sorry about that. Okay, so the question that we were looking to answer was specifically what the relationship is between MSAT concentrations at near road stations and the traffic levels and the distance to the main road. We also wanted to know how these concentrations compared to nearby urban sites, and then what the health implications were of the MSAT measurements. And we did that by comparing to a number of chronic and acute health benchmarks, which I'll talk about a bit more in a second. And so the MSAT data that we used was acquired from the EPA air quality system. And we used either co-located or nearby wind data to isolate the times where the wind, the monitor was being influenced by winds that were coming from the main road itself. And we also took into consideration the percent completeness, so annual, how much data was available and whether it met the completeness thresholds, as well as for the increment measurements, we needed to make sure that there was a nearby urban site that we could compare to that was comparable and was also measuring the same species at the same time. And so once we've done all that filtering, it takes us down to 17 near road sites that we had available to look at concentrations directly. And then 13 of those sites were available for us to look at increment values. And so these two plots here are showing how many stations were measuring a given MSAT species. And you'll note that on the increment chart that only 2 stations were available to measure acrolein, and then 1 for benzoapyrene, and unfortunately none for naphthalene. And so we looked at the concentrations in a variety of different ways, so daily, annual, and 3-year average concentrations. And I just want to note that These stations are located all across the US. They're different distances from the road. And so this really, the approach that we've taken here is not akin to a true gradient study, since the traffic patterns, the volumes, the meteorological conditions, they all vary from site to site over the same analysis period. So that's just something to keep in mind when I go through some of these results. And so I'm first going to walk through the concentration data with respect to the distance of each station to the main road. And the traffic volume that's indicated by this AADT value. And so here we're looking at the average daily values from each monitor across the 3 years of data that we have available. And so first we'll start with acetaldehyde. So those concentrations ranged from 1.1 micrograms per meter cubed to 2.07. And overall, as you can see from these plots, there's really no discernible trends with distance to road or AADT. If we look at the next species, which is benzene, we can see that the lowest concentration was in Pleasanton. The highest concentration actually occurred across 3 different sites, so San Jose, Columbus, and Providence at 1.1 microgram per meter cube. And you'll notice on the plot on the left-hand side here that these 3 locations are sort of spaced very there's a wide range of spacing between San Jose, Columbus, and Providence from the mainline road itself. But we still don't see any strong trend with distance to road or AADT in the benzene data either, which is interesting. The third species is ethylbenzene, and that ranged from 0.12 to 0.74. And similarly to benzene and acetaldehyde, there's really no trend that we see with distance to road or with traffic volume. If we go to the next species, though, which is 1,3-butadiene, we do start to see a trend. So overall, the mean concentrations ranged from 0.016 up to 0.25. And something I'll point out is despite having the highest AADT values, so these, if we look at the right-hand side map, you can see the values at the lower right-hand side of the plot. Those are all located in California. So those are Pleasanton, Berkeley, Oakland, and San Jose. So those have the highest AADT values, but we're an order of magnitude lower in the 1,3-butadiene concentration than the near road sites in other states. And that sort of is leading to this decreasing trend with AADT. And so the reason for this is not immediately clear from the data set or any investigation that we were able to do into the data, but there's a possibility that it could be due to different fuel formulations or other regulations that are present in California. If we look at formaldehyde, we see that there's a positive correlation here with increasing distance to road, so that's on the left-hand side. And there's a weaker correlation with AADT, so traffic volume, which you can see on the right-hand side. And so at first, that might actually seem somewhat contradictory, that there's, contradictory trends here. But the stronger correlation is with the distance to the road. So this one would be the one that we would sort of have more reliance on as being more reliable. So overall, what are all these different plots telling us? Well, overall, the near road findings suggest that the concentrations were really being driven predominantly by the urban scale background rather than the near road or the roads themselves. So then we'll switch gears. So next we wanted to look at how the concentrations compared with nearby urban monitors. So we compiled daily concentrations at each near road location and concentration at a nearby urban site. And we calculated the increment, which is shown here in this equation. It's the difference between the concentration at the near road site and the concentration at the nearby ambient site. And then we aggregated those results into 3-year annual averages. And since each MSAT had a different number of sites with data, we aggregated the results together by MSAT species into a box plot to better understand what MSATs are consistently higher at near road sites compared to the nearby urban sites. And so that results in this box plot here. And so what we can see from this is that the MSATs that were significantly higher at a majority of near-road sites for the following species. So we have for benzene, for formaldehyde, and for acetaldehyde. But the other species that we have here, so the other three, those increments were actually indistinguishable from zero. So if we take those three species that showed a significant increment and we compare them to the distance to the road and the traffic volume data, we get the results in these plots here. So these plots, just to orient you, the darker markers are the significant increments, and the paler color are the ones that aren't significant, but we included them for completeness. But they weren't used in creating the line of best fit, which is the gray dashed lines. So if we look at these, we can see that the significant increments actually have little correlation with distance to road or the traffic volume. And so this might seem surprising at first, right, given what I just showed you on the previous slide, which is that they showed significantly higher values. But we have to keep in mind that this was not a perfect comparative study, as each site, again, had a unique set of conditions, traffic volumes, and meteorology. And so while the previous slide showed that there's a statistically significant increase of these species near road, given the non-ideal comparison of the sites to one another, we actually might not expect them to show obvious trends with distance to road or with traffic volume. But nevertheless, they were, significantly higher or significantly statistically higher, it's a hard thing to say, at the near road sites. So next we get to sort of the most important part really is the, comparison to the health thresholds. And so we compared the near road concentrations to chronic cancer and non-cancer benchmarks and to acute thresholds, but I don't have time to talk about the acute threshold results today. And we used a variety of health benchmarks for these comparisons, which include benchmarks from 3 EPA data sets, as well as agency benchmarks from California, Texas, Virginia, and Washington State. And there are methodological differences in each of the benchmarks that we used, and that was to demonstrate the range of dose response values that might be appropriate to compare at a given site when screening for potential health risks associated with a given pollutant. And so first we'll look at the chronic non-cancer benchmark, which we're comparing to the annual means of the various MSAT species. So to orient you to this plot, the colorful markers are the data from the individual monitoring stations. The size of the marker, which is probably a little bit difficult to see, is the year. And then the gray markers are indicating a different non-cancer threshold for a given species. So what we can see from this plot is that the species that were well below the chronic dose response thresholds were 1,3-butadiene, acetaldehyde, benzene, benzoate-pyrene, and ethylbenzene. There's also acrolein concentrations which were at or above thresholds for EPA, California, and Virginia benchmarks, so the sort of a spread of results there. And also for formaldehyde, the concentrations were below the California, Texas, and EPA benchmarks, but near at or above the Virginia benchmark. So it really depends on which threshold you're considering as to where some of these values fall for certain species. If we, however, look at the cancer benchmarks, we see that all of the aggregated annual mean concentrations were well below the chronic cancer thresholds here. So we're well clear of these values. That brings me to my summary. So neurobenzene, acetaldehyde, and formaldehyde concentrations were higher than their nearby urban concentrations at most of the sites that we looked at. 1,3-butadiene and ethylbenzene increments were indistinguishable from a value of 0. The MSAT concentrations and increments did not correlate strongly with AADT or the distance to roadway in most cases. 1,3-butadiene, acetaldehyde, benzene, and benzoate-pyrene, and ethylbenzene were below the non-cancer threshold, and formaldehyde and acrolein showed mixed results. But one thing I should note is that acrolein is sort of termed acrolein unverified because the method with which we have available to us to measure acrolein is actually not verified by EPA. It's sort of it's the best that we have available, but it's not the best that it possibly could be. So you have to take the acrolein results with a grain of salt. But finally, all near-road MSAT concentrations were consistently below the respective cancer health benchmarks. And with that, I will thank you for bearing with me through my technical difficulties and for listening. And I'd be happy to answer any questions.
Great. Well, thank you so much, Olivia. And well, thank you for bearing with it and trying to figure all of that out at the spur of the moment while you're in the spotlight. So thank you, and appreciate the audience as well. So I use the EPA's data set quite a lot, and it seems to me that your results have some implications for them in the way they do the modeling that develops that data set. Have you thought anything about how you might translate what you've learned, and kind of inform NADA going forward?
That's a really good question. So I don't know that we've thought too much about how this data could be used moving forward, but we are trying to put a publication together on the results from this, which are obviously longer than the time I had to explain them today and more involved. But yeah, if we put that together, then I think that could be something useful for people moving forward to be able to use, to make changes to the way that things are done.
Great. Okay, I'm not seeing anything in the Q&A, so I'll thank you again, Olivia. And please, anyone, if you do have questions as we move along, please type them in, and we can always circle back. So, Andrew, are you ready? Great, we've got your slides.
Slides should be up there.
Very good. Take it away.
Great. So thanks, everybody. Today we'll be talking about benzene exposure and cancer risk from commercial gasoline station fueling events using a novel self-sampling protocol. This work was published a couple of weeks ago, and the handout should be a PDF of the paper. So first thing we want to talk about is what gasoline is. It's something that we all tend to be in contact with fairly often, probably. And it's a non-uniform hydrocarbon mixture. And we say non-uniform, it is very non-uniform. This table on the right is out of date by about 2 decades. But in general, the idea that the composition of gasoline is not a percentage point here and there. They can be 10, 20, 30 percentage points difference by volume of these various compounds. So it is definitely not something that is 100% the same across gas stations, time of year, states, etc. Since the late 1990s, ethanol has been added to gasoline from about 10 to 85%. E10 or E15 are the most common. If you go to a gas station, the little E number you see on the pump, that indicates what percent of ethanol it is. Important to note that gasoline is regulated on average, not a per barrel basis. You can imagine that testing every barrel that comes out of a refinery is not possible. So it's on the aggregate. And gasoline in the United States is likely to contain between 0.5 and 2% benzene, with the regulation being, I believe, 0.62%. And what are the health effects of benzene exposure? We know that benzene is toxic, as many people at this conference have mentioned, and it is strongly linked to leukemia, both occupationally and non-occupationally. IARC defines it as carcinogenic to humans, so a class 1 carcinogen, which is the most potent group. And the acute myeloid leukemia, or AML, is the cancer that has the strongest association, although there's some other potential associations with other forms of leukemia. And the carcinogenicity is a result of reactive metabolites, such as O-benzoquinone and P-benzoquinone, you can see at the top there. Who is exposed to benzene? Pretty much everyone. It's an ambient pollutant, as mentioned previously. The National Air Toxic Assessment data includes it as one of its list of contaminants. And between 20 to 40 million people per day are exposed to benzene specifically via gasoline vapors at gasoline stations. There are about 360 million gallons of gasoline dispensed per day in the United States as of about 2015 or so. Interestingly enough, consumer exposures, so that would be people who go to gas stations to purchase gasoline, snacks, drinks, etc., have not been investigated in the United States for nearly 30 years. And so we decided we want to take a look at this. And how do we measure exposure? We took 34 Baltimore area consumers into our study. This was originally supposed to be about 100, but we were recruiting and conducting the study in March of 2020. So that kind of got ground to a halt sample size wise with the advent of COVID. One thing we did that was pretty interesting was because of the difficulties associated with getting a single gas station to allow us to come in and sample everyone and everybody, we allowed the consumers to pick their own gas station. We gave them a backpack which contained an evacuated steel canister and essentially an on-off switch on their sampling line, so they could and were instructed to go to the gas station of their choice, do everything normally, and when they got out of their car, open their canister, and when they got back into their car, turn their canister off. A couple of things that the benefits here are that it avoids the simulation study issue where you have 6 scientists staring at somebody while they do something, which is a little unrealistic for the real world. And also they can fill at the gas station of their choice at the time of their choice, so you get a nice variability in what's going on. What were the results of our study? All the samples were 6-minute maximums of our canisters, and unsurprisingly, were far below relevant occupational exposure limits. Benzene PEL is 1,000 ppb or 1 ppm, and the benzene REL is 100 ppb. And you can see based on our violin charts on the right that pretty much everything for benzene was below 20 ppb, with I think one outlier in the 40s. And it's important to note that we have these exposure values. So what happens when we conduct a risk assessment, and not just a risk assessment, but a probabilistic risk assessment, because we have these distributions of values. We did this in a 2-pronged approach. We did the consumer and then an occupational. Consumer is somebody who goes to a gas station a few times a week, pumps their gas and leaves. Occupational would be someone who works at a gas station. In particular, we were thinking of pump attendants, such as those in New Jersey and parts of Oregon, that are legally required to pump your gas for you and may be exposed to gasoline pumping activities, for an 8-hour day. For both the consumer and occupational scenarios, we use the same concentration distribution. This was from the sampling we did for the consumers. The fill frequency, duration, volume for the consumers was distribution was obtained via questionnaires we asked, and the unit risk was obtained via the literature. For the occupational group, frequency of exposure, duration of exposure, it's all taken from the literature and kind of our professional judgment about how many days a week someone might work. So again, in general, at the bottom, here's our risk assessment equation. And this was essentially the same for both groups, just with different values for the variables. What are the risks to consumers? Unsurprisingly, we found that 0.0% of risk values for consumers exceeded the risk management limit of 1 in 1 million, excuse me. The x-axis here is log 10 of excess risk, so we are generally speaking not even really approaching something that would be exceeding the risk management limit. And this is for consumer scenarios where there's a wide distribution of sampling times, sampling duration, exposure durations, exposure frequencies, etc. And then for pump workers, these would be the individuals who work at gas stations that are out in the pumping area, pumping gas for people, not, for example, somebody who works behind the register. We found that less than 0.01% of values exceeded the risk management limit for our Monte Carlo probabilistic risk assessment. And again, this is a 1 in 10,000 excess risk management limit. So in general, for both the occupational and consumer scenarios, we found that both were below what we consider to be the risk management limit. But it's important to note that, again, we drew it back to the beginning that benzene is an ambient pollutant. Does your short duration pumping exposure when you go to buy gas cause equivalent risk to your 24/7, 365 ambient exposure to a lower concentration of benzene? So the way we did this was where we took the NATA exposures and the NATA risk values published by the EPA and essentially made a ratio. So the risk of the distribution of pumping risk for a consumer divided by the ambient risk for consumer based on where they lived in Baltimore. And again, you can see that little red arrow here all the way out to the right shows that the log 10 of this ratio, which would be 1, if it was equivalent, so we are far in the negatives here. So the pumping risk exceeds the ambient risk for 0% of consumers. So essentially there is no additive risk or a very minimal amount of additive risk for consumers pumping gasoline compared to what they were exposed to in Baltimore on an ambient basis. In an occupational setting, we did the same thing. We found the pumping risk exceeded the ambient risk for 29% of workers in the Baltimore area. It's important to note this is the Baltimore area because the Baltimore city has in the 95th percentile of ambient benzene exposure. You could imagine if this was in a lower populated or less urban environment, these distributions might change because the ratio of essentially how much ambient risk there is might be significantly decreased, or could be increased if they were in various parts of New Jersey and New York. The primary takeaways from this are that self-sampling using backpacks, for example, as we did is a possible methodology for collecting samples that would be very difficult to acquire otherwise for environmental behavioral reasons. The downside, obviously, is that we weren't able to supervise anyone. So we don't actually know that they followed the rules of the protocol beyond checking the vacuum of the canister. And the takeaways for the consumer exposures are that gasoline pumping activities do not approach the relevant excess risk management levels, and risk from ambient exposure for consumers in the Baltimore area is greater in all circumstances. And again, for the occupational group, the pump workers, gasoline pumping activities do not exceed the relevant excess risk management levels, and the risk from ambient exposure is greater in about 70% of circumstances. So again, there's about 30% of the workers in the Baltimore, 30% of possible worker scenarios in the Baltimore area where the pumping or the pumping risk exceeds their ambient risk. Acknowledgements, Dr. Kirsten Kohler, the advisor for this work, Dr. Mary Fox on the panel, Dr. Misty Levy-Zamora, Department of Transportation in the NIOSH ERC, as well as CARTI in the Johns Hopkins Bloomberg School of Public Health. We have a question from Olivia. We recruited the participants the same way everyone recruits participants, emails and flyers. I will say that we had some bigger schemes in progress for recruiting, again, that were put on hold from COVID. The participants are provided with results afterwards. The gas stations were not, because no gas stations themselves were actually contacted or used in the work. We don't identify the gas stations, and we explicitly said to our consumers, do not identify the gas station you got gas at. So we don't know and wouldn't be able to do that.
Right, thanks, Andrew. And just to give a little bit of extra context, because I've been continuing to look at this data set and sort of taking it into the cumulative risk analysis as I presented a little bit earlier today. I don't know, hopefully some of you saw that talk as well, and so you'll be able to kind of link these two pieces together. But the excess cancer risk in the Baltimore area, and we did draw participants from sort of the greater Baltimore, DC metro area, although predominantly Baltimore. It's about 35 excess cancers per million over a lifetime. So it's still a very low risk from the ambient air as well, but it does exceed, sort of our rules of thumb for in environmental health, what we become concerned about and want to look a little bit more closely at. And Andrew, I don't know if you wanted to talk a little bit about the design of the sampler, and kind of we had a little bit of a couple of hiccups early on.
Sure, yeah. So this was Chubb Environmental helped us quite a bit with the design of the sampler. It was fully custom, which was nice, in the sense that we were able to get this design kind of explicitly for what we needed for our role. The first version was we have this canister, it's a 1 liter, sorry, a 0.6 liter canister that goes into a little hiking backpack. and there's a silenite line that runs out to the participant breathing zone, so it ends up being right about here. Initially, we had issues where the gasket associated with the on-off switch kept sticking, and so we had scenarios where people had to literally manipulate the sampling orifice, which is not good. So we had one value that was something like 5 or 6 orders of magnitude bigger than all the other values, which would indicate somebody like bathing in gasoline. So we had to drop one of those. And then we had to redesign it so it's a gasketless design. So it's just like an on-off knob that the individual doesn't have to manipulate anything related to the orifice. But in general, it worked really well. People were able to follow the instructions. Again, it's an on-off and it's a little backpack. It's not very intrusive. It's not packed full of electronics or anything like that. So it wasn't unduly expensive or difficult to give to people. It was a tiny hiking backpack and a pound of equipment, and people, generally speaking, had no problems using it.
Great. Well, thanks again, Andrew. And if anyone does have a question, please enter it in the chat and we can circle back to it if we need to. And so on to Ranga.
I hope my screen is visible.
It's coming now, looks like. Okay, we've got it.
Okay. Yeah. Shall I begin?
Yes, go ahead.
Okay. Good morning to everyone present. I'm Ranga Rajan. I'm a PhD candidate from the University of California, Riverside. So today I'll be talking about the impacts noise barriers have on the near road air quality. So before we get into the noise barriers, I'd like to start off by explaining what transformation conformity is. So transformation conformity is required in the US under the Clean Air Act for any new transit projects that are being planned. So the main aim of transportation conformity is to ensure that these new projects do not lead to new or worsen existing air quality violations. So the general approach to doing this is to use a dispersion model to predict the impacts. So it's very essential that the regulatory models that are being used for this purpose are state of the art. So recently, noise barriers have become quite common in freeways in California. Although the main purpose of these noise barriers is to reduce noise pollution near freeways, many recent studies have shown that these barriers might have a mitigating effect on air quality nearby. So we look at one such study here, which was performed by Heist et al. In 2009. It was a wind tunnel study. So the picture on the left shows the setup he used. So he used 6 line sources here to simulate a 6-lane freeway. So the winds were perpendicular to the line source here. So the top picture on the right shows the wind profile from the front view of the setup, that is looking at the setup from here. So we can see the 3 line sources and the barrier here. So the presence of the barrier here has 2 major effects here. So the first effect is we can see a recirculating zone that is formed behind the barrier. So this mixes everything below the barrier. And the second effect that we see here is the lifting of plume up to the barrier height. So the plume here is getting lifted up to the barrier height here, which can be seen here. So this lifting of the plume increases the vertical spread, which dilutes the concentration. So this effect can be seen in the plot below. So the top 2 plots here show the zeta here, which is the non-dimensional concentration as a function of distance from the barrier. And the bottom 2 plot, so the top 2 plots are the case when there was no barrier present, and the bottom 2 plots are the case when there was a barrier present. So we can see that there is more than 75% reduction in concentration when the barrier was present. So it is critical for dispersion models to account for these barrier effects, especially when being used for regulatory purposes. So the main motivation for the study stems from the fact that the current regulatory models do not include these barrier effects. So there's a need for a regulatory dispersion model that does account for barrier effects. So R-line is another model. It's actually it's currently under research and being developed by EPA. It does have an empirical formulation to account for the barriers, but this formulation has to be evaluated. So there's also a need for studies with real barriers that can be used for these evaluations. So this study here directly addresses these needs by conducting a tracer experiment under real world conditions with a real barrier. So the data from this can be used to evaluate dispersion models that account for barrier effects. So now we'll look at the experimental setup that we used in this study. So the picture on the right shows the top view of the study region here. So this here is a freeway. It's I-215, which runs next to UCR. So we used 8 vehicles fitted with SF6 tracer relay system. The red lines here indicate the lanes in which these vehicles were driven. So the blue line here indicates the noise barrier, which is present on the downwind side of the road. The noise barriers were the height of the noise barrier was approximately 5 meters. So the yellow circles here indicate the sample indicates the locations where we sample the air. So we use 6 back samples per location to do this. So each back sample measured for 30 minutes to get a 30-minute integrated sample. And these back samplers started measuring one after the other. So the total measurement period was 3 hours. So the wind direction is indicated by the white arrow here, and the wind direction was predominantly westerly. We also did some wind measurements using sonic anemometers, which were placed in the regions indicated by the red triangles here. So this study was conducted on 4 different days, targeting different meteorological conditions. So this slide here shows some pictures of the setup that we used. So this picture here shows the SF6 cylinder, the outlet of which is connected to the exhaust pipe here. This picture here shows the sampling box with the 6 pumps and 6 back samples inside it. And this here shows the laboratory setup we used to analyze the back samples we collected. So now we'll look at the barrier model that we used to interpret the data. So this picture here shows the source in the red triangle. So this is a line source which is going into the plane. So now this source will create a concentration distribution at some distance. And this concentration distance can be assumed to be Gaussian in the vertical, and this is shown by the red line here. So the concentration is represented by the vertical axis and the height by the horizontal. Sorry, the concentration is represented by the horizontal axis and the height by the vertical axis. And we can see that the concentration is reducing with height until it reaches the background. So now let us introduce a barrier here. So this barrier will have 2 effects. The first effect is the mixing of plume below the barrier height. So everything below the barrier height is mixed due to the generation of the recirculation zone here. And the second effect here is the lifting of plume up to the height of the barrier, which is indicated by the arrow here. So these 2 effects will lead to a concentration distribution that is represented by the blue line here. So below the barrier, because everything is getting well mixed, our concentration is going to be constant. And above the barrier height, because we have this effect of lifting of the plume, so we just so this here just follows the same distribution as this, but multiplied by a factor FQ. So this is here is the concentration without the barrier present, which is this red line here. And because this is getting lifted up to the barrier height, I am subtracting this by the barrier height, which is H0. So the below the barrier height, it is the same factor multiplied by the concentration at Z equals to 0. That is the concentration here. Gets fitted to this point here. So there's one more term here called Fe, which is the entrainment factor. So this entrainment factor becomes crucial, especially when the wind speeds are low. So when the wind speeds are low, this recirculation zone doesn't have enough turbulence or friction to entrain the pollutants behind the barrier. So this leads to a reduction in concentration. So we'll look at the effects of this entrainment factor in detail later in the talk. So this plot here shows the vertical profile generated in the wind tunnel study, and we can see that the profiles generated in the wind tunnel study are very similar to the profile generated by the model. So we can compute the scaling factor FQ by performing a mass balance between these two profiles here. So now we look at some results from the study. We'll first look at the meteorological data that we collected during our measurement period. So the plot on the left shows the wind speed during our measurement period. So you can see on days 1 and 2, the wind speeds were quite high, whereas on day 3, the wind speeds were consistently low. And on day 4, the wind speed started out high, but then became low. The plot on the right shows the kinematic heat flux that we collected. And although on days 3 and 4, the measurements were done during night, we still noticed that the heat fluxes were always greater than 0. I think the main takeaway from the slide is the wind speeds on day 3, which are very low. So this plot here shows the model performance. So the 4 plots on the left show the performance without considering entrainment, or without considering the entrainment factor FV. And the 4 plots on the right show the results considering entrainment. So the vertical axis here represents the SF6 concentration, whereas the horizontal axis represents the distance from the barrier. So you can see that on days 1 and 2, the model performs well whether we consider entrainment or not. But on day 3, when the wind speeds were very low, we see that there's a big overprediction in the concentration, which reduces by a huge amount when we consider entrainment. Similarly, on day 4, there's a slight overprediction, which improves when we consider entrainment. So we can see the entrainment factor becomes important when the wind speeds are low. So this plot here shows the concentration of the predicted to the observed ratio with friction velocity. So the friction velocity is generally proportional to the wind speed. So if the wind speeds are low, the friction velocity U star is going to be low too. So we can see when the U star is low, the overpredictions are quite high. The predicted concentrations are almost 4 times the measured concentration. So this plot here forms the basis for the empirical entrainment factor that we formulated here, which is shown here. So this entrainment factor will tend to 1 as U star becomes higher. So this is the same plot we see here, but I've included one more line here, which is represented by the green triangles here. So these green triangles are the predicted concentration when there is no barrier present. So we can see that there is at least a 50% reduction in concentration when there is no barrier present. And this reduction in concentration decreases as we move away from the barrier. So these are the 3 main conclusions from this study. So the first is that the presence of barrier results in more than 50% reductions, which are consistent with idealized studies such as the wind tunnel study we saw earlier. So this shows that these idealized studies form a good guide to reality. Also, this work is the first step towards including barrier effects in regulatory models. I think more detailed analysis is required before we actually reach that state, and I believe both Caltrans and EP are in the process of doing that. And current models are also highly likely to overpredict concentrations under low wind speed, and this overprediction can be reduced by considering an entrainment factor. So these are some of the future work that we have planned. So the first is to conduct a similar study in a site with no barrier, and we'd also like to perform the similar study with the site with barrier on both the sites. So the more no barrier case can form as a control for this experiment. The plot on the top right shows the velocity profile when we have 2 barriers present. So we can see that there are 2 recirculating zones generated here. So this might give us some interesting results. So finally, the entrainment factor also needs to be a function of downwind distance, because physically, as we move away from the barrier, the effect of entrainment will decrease. This can also be seen from the profile at a distance 15 times the barrier height. This profile is very similar to the profile when there is no barrier present. So I'd like to end this talk by acknowledging the people that were involved in this study. I'd first like to remember Kurt, who is no more with us. His contribution was very crucial to the successful completion of this work. This work also involved a complex field study and wouldn't have been possible without the involvement of these 18 undergraduate students. They were the ones who drove the vehicles, did the analysis of samples collected. I think someday they have to work the whole night to complete this project. I would also like to thank California Department of Transportation for funding this work. Thank you.
Thanks so much, Ranga. And yes, absolutely. Second your thanks for the students who who drove the truck, right? There's only so much we can do on our own, and we certainly appreciate the students. All right, are there? I don't see any questions for Ranga. So Ranga, you mentioned that you think that what Caltrans and EPA are doing additional studies.
So they'll be using the data set that we collected to like evaluate the models that they are working on. That includes the barrier effects. So I think the main objective of this study was to collect a data set that could be used to evaluate dispersion models that includes barrier effects.
So I wonder about the and how you feel about the generalizability. So I mean, is the work that's going to continue? While Caltrans obviously is focused on California, but are there any issues about sort of translating this data into other settings at all?
I don't believe like moving, for example, changing the location from California to a different place is going to create a big difference in the models. I think as such, the model has the uncertainties in the models would be much higher than the uncertainties that will be developed due to this movement.
Just in a different setting would not make that much difference. Okay.
I think even without that, the uncertainties would be quite high.
Great. Well, thanks so much.
Thank you.
And we're a little bit ahead of time, but that's okay. Get an extra few minutes to grab a coffee or something to eat before we come back for our next session and wrapping up the conference. I hope you all will stick around. Thank you for joining us.